Welcome back, and now we're just going to do the very last thing that we're going to look at in terms of DNA structure and inheritance, and that's gene technology. And this is how we can actually get a picture of the different alleles or the fragments of DNA of an individual. So this kind of thing is used for forensic testing, for matching DNA perhaps found at a crime scene with individuals that are suspects, but also for genetic screening to be able to tell if a person is perhaps a carrier of a disease trait, perhaps like cystic fibrosis that we've talked about, or other disease traits that you may want to screen for if you're worried that you are a carrier for a particular disease that would perhaps impinge on the life of your prospective child. So some people will use screening to get an idea as to whether or not they want to risk having a child if there's a high chance that they may be born with a particularly devastating disease. So we know from when we did DNA structure that DNA is a series of nucleotides that are put together with a phosphate, the sugar, the deoxyribose sugar, and the nitrogen base, which can be our adenine, thymine, cytosine, or guanine. Okay, so the key thing with our phosphate though is these phosphate groups are negatively charged, which means that along the backbone here on our DNA in this diagram here, this is actually really negatively charged. So what that means is overall DNA has a slight negative charge. It's not a really strong negative, but it's enough for us to be able to differentiate DNA from other things. So we can use a process called gel electrophoresis. Gel because it's actually done in a medium that's like a thick jelly, to sort DNA of different size fragments. So if I have my DNA strands, it gets chopped up into pieces, and then we can separate out those pieces using the gel. Okay, so DNA samples of unknown lengths are placed into little divots or wells. So if we have a gel like this, it has little wells that come down into it, and we can load the DNA into these little pockets in the gel. The gel is then sat in an apparatus that, if we were to look at it from the top down, so if we change our perspective when we look at it from the top down, the wells would be here that have got the DNA. We would attach it to a positive electrode here, negative electrode here, and there's a solution that it sits in that allows the current to flow. Because the DNA pieces are negatively charged, they will be attracted and move towards the positive electrode. Okay, remembering the opposite charges attract. So it will move from the negative terminal to the positive terminal. Okay, so DNA will then migrate from the negative towards the positive. The smaller the pieces, the faster they travel. Okay, so the smaller the pieces, the faster they travel. So small pieces travel quickly, so they travel further. So then we will get bands forming, and then the one that is closest to the positive terminal will be the smallest fragment. So this is a picture of a gel that's running up a line. So these are the wells here. This would be your sample DNA that is being placed in. This is your negative electrode or terminal. And then this is the positive terminal. So because the pieces are negatively charged, they will move down this way. So we can say that this piece is smaller than the piece up here. So this would be larger because the smaller pieces move further. So then we can compare the lines that are seen. So we can see here we've got these patterns of lines, where lines are in the same place, then they're the same piece of DNA. So this means that if we take DNA from the mother and the father, we might see a different combination of alleles like this. And then the child, we can see which traits. So if this is the mother, if this is the father, then we would expect the child might have some from the mother and some from the father, and we can identify disease traits and compare them. So let's have a look at this and try and make it a little less confusing. So this is what we refer to as a DNA fingerprint. Okay, this is what we get from gel electrophoresis. Okay, so what we see is the pattern of the pieces. Okay, so proteins chop up the DNA into lots of little pieces, and then we know where certain genes are. So it's basically a comparative one. Okay, so we can have a look here. We have the mother, the child, and then 3 males. Okay, so from this is sometimes how paternity tests are done. So if these are 3 potential fathers of the child, we can see which things are common from the mother and then which ones would be, remembering that the mother and the father are a combination. So we can see here that the child shares these ones with the mother. Okay, this one also with the mother and this one with the mother. Okay, this one, however, if we change color, this one, however, here is not present in the mother. So if we have a look at the other possibilities from the father, we can see it's present in this one, but not here or here. Same goes with this one here. This is in the mother, but doesn't turn up in the child. Okay, so that's not going to be one that's going to be diagnostic for us. But we can see here that we have this band here in this male and here that doesn't appear in any of the other males, and we have this one and this one. So from this, we would say that this is most likely to be the father because they have the most alleles in common between the child and the father, and a lot of the other alleles are coming from the mother. Okay, so we can see that this child is a combination of this mother and this father here, and then we can use that to help us identify the father. Okay, so we can see here these 3 here coincide with male 2, and we don't have that same similarity between male 1 and male 3 with the child. So male 2 is most likely to be the father. So what we can do is look at these in terms of some other questions. In this case, the question is a little bit wordy, so let's have a look at what ones tell us important information. This one states that gel electrophoresis, which is the process that we're talking about, can be used to separate DNA fragments in forensic chemistry. A mixture of fragments, and this is just the unit that we use to measure fragments, so kilobytes. So we can see this one is smallest than this one and this one because it's 0.55, 6.3, and 25. So is placed onto a gel. So 1 kilobyte is basically 1,000 bases. Kilobases is our measurement. We apply the electric current, so remembering that the DNA moves from the negative to the positive, okay, through the gel, and the gel fragments are stained and become visible as bands on the gel. The question wants us to label the negative and positive terminals of the gel and then label the DNA fragments according to their size. In order to be able to label the gel, we need to know where the samples were placed in. The diagram tells us here that they were placed along this line. Remembering that the positive terminal is opposite to where the samples were placed, so that must be here, making it the negative terminal at this end. Our DNA will now move in this direction. If that's going to be the case, remember the smallest pieces travel the farthest. So that means that this must be the smallest, then the next, and this will be the largest. So in this case, this must be the 0.55 kilobase fragment. This will be the 6.3 kilobase fragment, and then we have the 25 kilobase fragment will be the one that has moved the least distance. When we look at it, we can see that that is exactly what we see. Our largest will be here, smallest up this end, positive terminal. So that is it. Absolutely it for all the content that we need to do. And I will see you in class so we can start workshopping some solution to some of these questions.